So, very warm welcome to all of you, all students, for today's lecture. Each one of you, whosoever were with me yesterday. So today we are again meeting for the MBBT course, Molecular Biology and Biochemistry Technique. We will discuss now from today onwards some basic techniques that we use in the molecular biotechnology. Before we start ahead, it's always I keep first 10 minutes for your previous sessions. If you have any questions from yesterday or some kind of QA type of for 10 minutes, then we can start with the lectures as per the things are. So the first 15 minutes is to let everyone join our sessions. So I hope whosoever has been there, they have joined our sessions by now. And now we can start with the things. So today is day 2. So yeah, yesterday I was teaching you about isolation of DNA. So I missed the virtual lab for the same. First we do the lab thing, then we go ahead. Yeah. So how are you all? How was yesterday evening? Did you celebrate something? You went somewhere outside or spent some time with friends or family or something like that. I just got some jalebis for my family, so this is very traditional going on, and then we just ate in the evening. That's it. Nothing special. Mm. And those who has not subscribed to the channel, please subscribe and make it up. So thank you so much, whosoever has subscribed so far. I really appreciate all of you. I really thank you so much for your kind gesture. And yeah. So anybody has any questions from yesterday? All things were clear yesterday? And this time is good, right? 8 o'clock. We start sharp at 8. I send you link at 7:45. So I previously finished my class at 7:15, and then I have this 30 minutes break, and now I'm next here with the next class here. So okay, no questions. In that case, I would like to start the virtual lab of DNA isolation. So let's start our session with DNA extraction. How we can extract in a virtual environment. So we don't have at the moment, what do you say, to do things in practical. So our sessions will be divided in such virtual labs, some videos, some lectures, assignments, like this. We will contribute. We will go towards our sessions. So that's how. I'm thinking of making these things more exciting for you. So in this experiment, we need to isolate DNA from human test subjects. Uh, so let's start. So DNA need to be isolated from while the DNA testing, body identification, or from the analysis of forensic evidences. So all from these different states, we need to isolate DNA, for which we need to do our work. So DNA extraction is typically the first step in a longer laboratory process. So DNA is important part because DNA first needs to be purified away from proteins and other cellular contaminants, and we need to have inside the nucleus that is present about 2 meters of DNA is present within that nucleus. So we need to extract that also. So this is our nucleus. Inside the nucleus, our chromosome is present, and within this chromosome, our histone beads. So this is our DNA is finally present like this. So first we need to collect some cells from our test subjects. The skin on the inside of our mouth loses thousands of cells every day. So these are the cells we are here after. So let's get started. First we collect the cheek cells, then we burst open these cells to release DNA, then we separate DNA from proteins and debris, and then we isolate our concentrated DNA out of it. So in these four simple steps, our isolation of DNA is being simply covered. So here are the materials equipments you will need to purify DNA from cheek cells. So you need water bath, microcentrifuge, pipettes, micropipettes, some solutions, swab, some Eppendorfs. So all these materials. Warm water bath, microcentrifuge, buccal swab, micropipetters, sample tubes, lysis solution, concentrated salt solution, suspended buffer, ethanol, isopropyl alcohol. So we need all these materials for our isolation of DNA. So let's collect the cheek cells by using the buccal swab, like we do it in the case of COVID test also the same way. So we just took the normal buccal cells from the cheek cells from buccal swab with the help of buccal swab, and now we will place the swab into the Eppendorf tube, and we have lots of these cheek cells residing there, and we have a lot of nucleus of each cheek cells that we have to extract. So we put them in the Eppendorf with the lysis solutions to break up, so that we can break upon uh, our cell membrane. So lysis solution, lysis means to break up, and we will add this to our lysis to the Eppendorf. So that's it. So place the tube into the warm water bath. So the cellular membrane is broken apart. You have your nucleus. So these are histones. Histones are also broken apart. So in the lysis solution, uh, we have two important things, that is detergent and enzyme called protein kinase A. Detergent helps to disrupt the cell membrane and nuclear envelope, causing the cells to burst upon and release their DNA. Where DNA is still wrapped around these histones, for that protein kinase A cut these apart to free the DNA. So his protein kinase K will help to break apart your DNA, your histones from your DNA, and detergent will help to break apart your cellular membrane. So cells have stayed in the warm water bath. So now we brought it outside. Now uh, we will remove the swab, and we will add our concentrated salt solution. So concentrated salt solution is added now. So this salt solution, what it will do, it will cause proteins and other cellular debris to clump together. The salt that we have added, salt solution, it will clump together all the debris and proteins together and separate the DNA from it. Then we'll place these tubes into the centrifuge. So in order to make it balance, we will add another Eppendorf on the other side of equal volume, so that balance should be maintained. We will close the lid and turn it on. So once it is done, so we will see protein and debris to be at down bottom and DNA liquid to be present and up in this way. So the heavy clump of debris and proteins, they come to bottom of the tube, while the strands of DNA remain distributed through the liquid up. So we will take carefully the supranatant from there, DNA, and add into other append off. So now it's time to isolate concentrated DNA with isopropyl alcohol. So add some isopropyl alcohol to the tube. So what will this isopropyl alcohol will do? As in alcohol, DNA is not mixed up, so it will get separated, and we have come out of the solution, and now we can see clump of DNA with our naked eye. So our DNA came out from that solution, and we can see it very nicely there. So we will centrifuge it again so that DNA could sink down, and we can have it, keep it for longer time in the cold for longer term. So we extract the liquid, and the rest at the bottom that you have is now DNA, and you can keep it in minus liquid nitrogen minus 197 degrees Celsius or minus 4 degrees. Yeah, it's up to you. If you want to use it soon, then keep it at the 4 degrees Celsius. If you want to use it for the longer run, then keep it in the minus liquid nitrogen for the longer run. So that's how you extract your DNA. That was simple virtual lab for the same. So I share this with you so you can have it later. So all the links that I'm sharing, please go through them. So all the informations are present related to your work things. Then just go through them once, and you can have the knowledge about it. So that was about from the some little bit about from the yesterday's work. So today's work is cell culture technique and microscopy. So we will touch these two parts of today's lecture. So let's start. Any questions, students? There are principle of genetics. Same gene, different organism concept. So like we discussed yesterday, so they took spider web, the genes that were producing web, they put that into the yeast organism. So yeast got multiply, and we can, so the process what happened is then we can extract you can extract um, the spider web from the yeast cells. So we were producing in a very large amount. Same way, this green fluorescent proteins GFP, they are being produced by the jellyfish, and we took that gene and let it into our organisms like bacteria or mouse cells to see things in more advanced version, so that we can analyze things in better way. So that's how these two things are really helping us to know, by having the same gene, GFP same gene, and the gene for making spider webs and producing into different organisms, and we can have the function of it in large amount. So that's how you make it. So, Bache, next question, is the process more or less same for extraction? So, yeah, I mean, in plant, you have to mince your plant samples in very small form. In animals, also, your tissues has to be very minced with that. Or if you have animal cell culture, then in cell culture it's different, but more or less it's same. It will be same. So there will be some changes for the blood. If you want to extract DNA from blood, it will be different, but mainly the main principle what we were seeing that so far, it will remain as it is. So during isolation of DNA, we use phenol chloroform mixture. So what is the function of these reagents? So alcohol, mainly alcohol reagents, they help to, as we discussed in this virtual lab also, they help to because DNA doesn't dissolve in it. So it helps to clump our DNA, get separated from that liquid, and we can remove our DNA easily afterwards. That's how we do it. That's why we separate our things and then we centrifuge it and then remove the liquid, and we have DNA at the bottom as a precipitate at the upper end of tube. So RPM could be 1200 RPM, 6000 RPM. It depends upon the protocol. So these protocols could be different from different labs. Do I have a protocol? Did I share with the protocol so far? Isolation of DNA. These were the lectures that I was teaching at the Mata Gujari College in Fatehgarh Sahib in Punjab. So I was teaching this DNA isolation, DNA extraction subject, and so this was a specific protocol. So before I go ahead with this, let's watch this video that will clear some of the basic concepts of yours that you're asking, why we are using these things. So this will clear the basic. I want to just place this video then.
Stated Clearly presents What is DNA and how does it work? DNA, also known as deoxyribonucleic acid, is a molecule. It's a bunch of atoms stuck together. In the case of DNA, these atoms combine to form the shape of a long, spiraling ladder, sort of like this one here. If you ever studied biology or saw the movie Jurassic Park, you probably heard that DNA acts as a blueprint or a recipe for a living thing. But how? How on earth can a mere molecule act as a blueprint for something as complex and wonderful as a tree, a dog, or a dinosaur? To help answer that question, let's first take a quick look at amino acids. Amino acids are tiny little chemicals inside our bodies that are so important, they're often referred to as the building blocks of life. There's about 20 different kinds, each with their own unique shape. The neat thing about them is they can be attached to each other kind of like Legos to produce an endless variety of larger particles known as proteins. Amino acids make up proteins. Proteins, along with other chemicals, combine to form living cells. Cells make up tissues. Tissues make up organs. And organs, when they're all put together and functioning, of course, combine to form living creatures like you and me. These proteins that make up our bodies, and keep in mind there's millions of different kinds of proteins, they each have to be formed in the perfect shape in order to function. If they're the wrong shape, they usually won't work. That's where DNA comes in. DNA does a lot of interesting things, some of which we don't fully understand, but one of its main and most well understood functions is to tell amino acids how to line up and form themselves into the perfect protein shapes. In theory, if the right proteins are built at the right time and in the right place, everything else from cells to organs to entire creatures will come out just fine. This here is a simplified model of DNA. It shows us that the steps of the ladder are made up of 4 different kinds of chemicals shown here by different colors and letters. If you look at just one half of the molecule, you can read its chemical sequence or genetic code from top to bottom, sort of like a book. A single strand of DNA is extremely long, millions of letters long. It spends most of its life coiled up like a noodle, living inside the nucleus or the centerpiece of a cell. Amino acids, however, live outside the nucleus in what's called the cytoplasm. To help DNA interact with the cytoplasm and convert those amino acids into proteins, special chemicals inside the nucleus make partial copies of the DNA code. These partial copies, called RNA, look a lot like DNA, but they're shorter, of course, and they're missing one of their sides. Their small shape and size allows them to fit through tiny pores in the nucleus, out to the cytoplasm, and into the mouth of another particle called a ribosome. Ribosomes are protein-building machines. They read the RNA code 3 letters at a time, suck amino acids out of their surroundings, and stick them together in a chain according to the RNA code. As the chain grows, it bends, it folds, and it sticks to itself to form a perfectly shaped protein. Every 3 letters of the RNA code tell the ribosome which of the 20 different kinds of amino acids should be added next. For example, CAA tells the ribosome to grab a glutamine, AGU tells it to grab a serine, and so on. Once a protein is built, it can then go on to do a number of different things, one of which could be to help form a brand new cell. So to answer the original question, what is DNA? DNA is a molecular blueprint for a living thing. How does it work? DNA creates RNA, RNA creates protein, proteins go on to form life. This entire process, as complicated, as sophisticated, as magical as it might seem, is entirely based in chemistry. It can be studied, it can be understood. I'm John Perry, and that's DNA stated clearly. Thanks for watching. If you liked what you saw, make sure to give us a thumb.
So that was a small story about what we did yesterday, a little bit more about DNA, how things are prevailing in the field of cytoplasm and how they are arranged very nicely. So very nice video about the same. So that's how our DNA is coiled, as we discussed. You have DNA and then histones, then centromeres, you have a chromosomes, then inside chromosome of nucleus is a chromosome present, like that. So this is one of the, like in the lab, if you enter, this is one of the protocol that they will give you, that you have to follow this protocol for extraction of DNA. And basically you can use it for any things, plant or animal. Is just you have to extract DNA. So that's you harvest the nucleated cells first, then lyse the cells, nuclei, digest the denature of the proteins, separate contaminants from the DNA, precipitate the DNA, resuspend DNA in final buffer that you are looking for. So this is how so we use those lysis solution, all these things, but in this case we are using these specific things to go for, specific chemicals to use for. So follow these steps, and then hence you will have your things to be done clearly. So I will share this with you, this protocol thingy, and so where was it? I hope also. So somebody was asking about protein. I hope that is clear. Yes, you can extract cancer cells and healthy cells, and you can compare between these two cells to check on, run on the agarose gel how the DNA is different from the cancerous cells and the healthy cells. You can check many things with that, with the help of that. So lukewarm water helps to do your reactions in very fine manner, because at the room temperature we have 20 degrees Celsius, then the warm water would be around 60 to 70 degrees Celsius. So at that reactions goes normally faster for the reactions. SCID infected cells for isolation. What are SCID infected cells? I have to look for severe combined immunodeficiency. Yeah, why not? You can extract DNA from all any types of cells and compare with the healthy cells, and you can create a new data out of it at the end. So I provided you the PDFs already in the presentation, so you can maybe copy and create a PDF out of it later. So I've shared you one PPT for the same, so you can go through that. So this is I shared principle of this PPT. You can get that copy and create a PDF out of it. So carbohydrate content is the problem while extracting the plant leaves. What can we do during the case to maintain the purity of DNA also? Uh-huh. To maintain the purity, check from the OD that how much protein is present in it. You have to do not have to have protein in it, so you have to do some reactions to do so. I'm just out of my mind with about that at the moment, and you have to check that there is no impurities are in your DNA that you also have to maintain. Um, yeah, that's true. There is a lot of carbohydrates, so you have to add one reagent. So anybody knows what was it? I am out of mind at the moment. I cannot remember it. So this was done. Okay, students, run on the gel. Joyson, what do you mean by run on the gel? So there is student asking pre-asking us questions that there will be problem of carbohydrate content. So what we can do during to maintain the purity of the DNA also. So you can of course you have to check by the OD how you can do it. So once I remember what I shared with you yesterday. So you can check the purity if it is more than 1.6, then it is a protein contaminations. If it is sorry, less than 1.6, if it is more than 2.0, then it is a chloroform phenol contaminations. So you repurify your samples actually. That's how you do it. Okay. So, so this is how we do it. So students, it's we already went a lot of time on it. So I want to get on with the today's lecture. I haven't started yet with you. So basically I will start first with the microscope basics. So let's start with today's lecture with microscope and cell culture. So first, the basic microscope. We don't have microscope at the moment, but still we can study about microscope. Those who have never studied in their life, for them it's a golden chance. You can have a great opportunity to learn the basics about the microscope with us now. So microscope, it's mainly magnifies objects, makes objects look bigger, and it helps to see things which we are not able to see with with the naked eyes. Um, so they are body tubes. So this is the body tube on the top, and you have occlusions on the top of it. You have a nose piece, objective. This is objective lens. Then stage clips are there, diaphragm at the bottom. Then there is a light at the bottom. Then fine adjustments, core adjustments. Stage is there, arm is there. There is a base. So always carry microscope with the one hand on top and other hand on the bottom, so that it's on the safe side. So there are three types of microscope: simple microscope, compound microscope, and electron microscope. So simple has one lens, compound microscope has two sets of lenses, and it can magnify around 100 to 200 times larger than they really are. And whereas electron microscope can magnify objects up to 300,000 times. And they do not use lenses but use electrons to enlarge the image. So let's start with each part of the microscope. First is ocular lens. This is the lens that we look through at the top. Then comes the coarse adjustments. There's a knob on there on the right hand side of yours. So you can turn the bring object into the focus with the coarse adjustment. Then the fine adjustment is the small knob that brings the image into more focus. So this is coarse adjustment, this is fine adjustment, and this is the ocular lens. Now comes the arm. Here, this is your arm where we're sporting the body tube. The part that holds the eyepiece and objective lens, that is the body tube is there, that holds eyepiece and objective lens. Then comes the nose piece at the bottom, the part of the bottom of the body that holds objective lenses and allows them to be turned on. So high power objective lens. So the lens that magnifies object 2 to 40x. So here within it there will be 10x, 20x and 40x. Three different kind of lenses will be attached to it. Low power lens would be of 3x and 4x, and middle power would be of 10x. So all threes are maintained together there. The flat part below objective lens where the slide is placed. So this is the flat part at the bottom. Stage. Then the clip is the ones to hold your slides, which doesn't move. And then the diaphragm that controls the amount of light that is coming from bottom. So control it, you can control it while entering the view of it. So light source, there should be a light source from the bottom, that is lamp under the stage that sends light towards the object being viewed. So that is the base that supports the rest of your microscope. So we have discussed in details about the microscope, what is what, from the ocular lens to nose piece to the various lenses, coarse adjustment, nose piece, fine adjustments, and light source, and diaphragm adjustments, base tube, side tube. So we have discussed all these things. So let's go one more time. Top is your ocular lens, left is your body tube now, then comes the nose piece, objective, stage clips, diaphragm, light, arm, stage, coarse adjustment, fine adjustment, then the base. So this is all together makes your a proper nice microscope. So this is the field of view, that's what you see in there. So whatever that you are seeing is there, you're looking through the eyepiece. You're looking through the eyepiece is the circle, in the circle form, and it will be opposite and other way around. So if something is seeing at 7x like this, So 10x would be like this. So it's better, much more magnified. So which of these two images from the left and right is higher magnifications? This one or this one? I think left one, or no, it's the right one. So here you can see that the image that is being shown here with these clump of masses being largely done there, that on the right hand side. So there are also power of your microscope. So to calculate the power of magnifications, multiply the power of ocular lens by the power of the objective. So you multiply the power of your ocular lens by the power of your objective lens. So like this, you have 10x, this is 40x. So you can calculate by just multiplying it. So find the power of the lens, it is found on the side of the lens, and magnification of a lens is always identified at the label of it. So multiply the power of the eyepiece by the power of the objective lens. So whatever the power of the eyepiece is there, you just multiply by the objective lens. For example, eyepiece has 10x and objective lens is 100x. So your and one thing is you have 10x eyepiece and 50x objective lens. 10x times 40 and different 40x objective lens. So each will have a different, so you just have to multiply 10x by 100x, so it will be 1000x will be magnification, 1000x. This will be 500x, this will be 400x magnifications. We just have to multiply that by power of the eyepiece and the objective lens. So objective appear to be upside down and backward. We see the back side of your lens of your object and it's upside down. Movement appear to be in the direction that is actual movement. So like this, E, 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 E. So whatever the direction of this was, it will be in this form, opposite, upside down and backward. So let's turn on the microscope, then rotate the nose piece to click that red banded objective into the place. So this is done in the practical sessions. This we can't do it now, but I want to give a feeling that whenever you work with a microscope, how things will be working out for you. So these were done in the microscope. So students find out. Okay, leave it. So how to make a wet mount slide. So whenever you're in the lab, you have to check any specimen that you're looking for. So you have to create on the slides, and how to create it. First you clean it with a dropper, the center of the slides, then use tweezers to lay specimen on the drop of water. Then you lay a drop of water at the center and then gently touch the cover slip to the edge of the drop of water to cover the specimens in the water. So you take we take it at the bottom and you just slide it down and check for slide it down and you just watch your things that you're looking for. So that's how you clean it. Put the specimens with some oil in it, and then you put your cover slip, make a 45°C angle, and then you put that cover slip on the top. So this way you can see the your results. So thank you very much. That was a simple technique about how you can check um, various things. So this is an example mutant genes. So these are three genes, pair of three legs. Just saying, oh, he saw mutant genes in there, but it's actually a joke, funny joke. Haha. So that's was something about basics about microscope. I hope you all might be aware of it, but I feel like if we are studying about microscope, basic was quite required so that we can go to the advanced version of light and electron microscope. So let's grab your questions. By X-ray crystallography, NMR, you can do many techniques uh, to check if a protein is hydrophobic or hydrophilic. So you have to go for higher techniques to do so. How can I explain how to create? Bache, I have discussed it yesterday in quite details. So students, okay, so far so good. We are on the right track. Let's start with the light and electron microscope. After this, this is finished. We will take some break and then we continue afterwards so that you can refresh a bit. It's not so much pressure on you also, and then we can start again. So let's start with the new topic within the microscope, light and electron microscopes. So this is the replica of a Leeuwenhoek microscope that he constructed in 1670s. It's present in the medical library of Texas medical branch, Galveston, Texas. And this is a replica of a Culper tripod. Culpeper tripod microscope built in 1725 by Edmund Culpeper. And there's a collection of Moody Medical, the same present there. So you can see how nicely woody construction is done to the microscope. And this is a replica of Marshall microscope by John Marshall with the collections of Moody Medical from Medical Library. So today we in this topic of light and electron microscope, we will be dealing with the various types of microscopes and study how the principle works. So first is light microscope. Under this we will go through the bright field microscope, dark field, phase contrast, polarizing, DIC, epifluorescence, confocal laser, uh, scanning, electron microscope, scanning and transmission. In electron microscope, we deal with the two types, scanning and transmission. And then we deal with the specimen preparation for the light microscope, SEM and TEM for respectively. So microscopes have been essentially quite a major need for the biologist, so that we can confirm whatever we see at the chemical level, at the biochemistry level. We can be sure that what things are working, they are really working in the what do you say, in the right manner. We are going on the right track, because what we see from the naked eye in the microscope, like how the Golgi body is moving, how the mitochondria our movements are happening, how the ATP production is being done. But if we see them with our naked eye in the microscope, with the help of aid of microscope, that we can be confirmed that this is the regular signaling mechanism is happening by seeing things happening in real life. So that's what the real microscopes are really helpful for us. So light microscope, so this is a bright field microscope, the most common general use microscopes. Bright field microscopes are named because of the field is bright, this bright field, that's why they are bright field microscope. And objective viewed is dark in this case. It's a simple design, ideal for the bacteria and tissues. So we have binocular bright field microscope with movable stage could be moved, and dioptic adjustments, condensers, iris diaphragm, and these are used as clinical research students for the microscopes. So they have high numerical aperture, good resolutions, ideal for the stained bacteria. Unstained specimens have poor contrast in this case, simple design, and we have halogen light source in it. So these are some examples that how in the bright field images looks like. Stained blood cells in the peripheral blood smear. This is the flagellate trichomonas. Sections of the gut tissues and containing ciliated parasites. So basic design of bright field microscopes has been modified for special uses. Inverted microscopes on the right, allowing viewing of the cells in the flask. well plated the deep containers that do not fit into the objective lens. So this is how inverted microscope looks like. So you have light source from here, you have objective lens at the bottom, and you have eyepiece here being there. So you see your images like on this on the stage. So these are inverted microscopes. So further, dark field microscopes, they creates a darker background. Background is dark in them to allow the viewing of the small unstained objects such as motile bacteria that would be difficult to view in the bright field. Whatever the things that we can't see in the bright field, we can see it in the dark field microscopes. And the central portion of the light is blocked so that only oblique light strikes to the specimens. So we can see bright man specimen dark background, can see very small objects, but resolution has to be variable. High contrast, good for unstained live and motile specimens. So here this is the dark field images. We can see of algae hydrodictyon reticulatum viewed with a dark field microscope. On the right hand side is a leptospira spirochete viewed with a dark field microscope. So we can see things in very nice way with the help of this microscope. Then comes the phase contrast microscopes, which converts the differences in the refractive index in the specimens to difference in the image brightness. The central portion of the light is blocked in this case, creating a light from the condenser that illuminates the specimens. So they are also provide high contrast, good resolutions, good for bacteria, flagella, cilia, organelles such as mitochondria, and good for unstained or live mounts. So phase contrast microscope. So you have phase objectives here on there, and you have annular ring at the bottom. So that's how the images looks in the phase contrast with the image of the DIC image. We will discuss about DIC microscope also, which is on the right. So here on the left side is the unstained squamous epithelial cells observed with the phase contrast microscopes. So we can see differences in the refractive index in various regions of the cells for contrast in the images. So because of it's mainly depending upon the refractive index, the main principle. It's because of that we see a different part of the cell. Then comes the polarization microscopy, in which you have a polarizer on the top, you have specimen here, and you have a polarizer at the bottom also. So detect specimens that are bifringent, that is have characteristics of double refraction. Velocity of light refracted by substance is not the same in all directions. So substance is placed across 90 degrees Celsius, two polarizers. You have polarizer at the top, at the bottom to each other. So you can check bright image, dark background with these microscopes used for substances with highly organized molecular structures such as crystals, minerals, and we can check things in the quantitative manner also. So this is how the crystals looks like from the ground being viewed by the polarizing microscope on the left side from the Martian meteoroid. So on the Martian meteoroid, and the top right is a bright field image of renal epithelial cells containing fat globules. And the bottom right is a high magnification view of the same cells view with polarizing microscope showing typical Maltese cross. So we have Maltese cross in the fats of the polarized cells. Then comes the epifluorescence microscope, which allows the detection of molecules and ions within the cells to check the molecules that are present, and also the ions, various ions, sodium, calcium, potassium, pump, phosphorus, kinases. So fluorescent dyes absorb short wavelength of the light and emit longer wavelength. So barrier filters and a dichroic prism select the excitation wavelength that strike the specimen and exclude the excitation wavelength from the detector, allowing only emitted light to reach the detector. Use UV light source, mercury or xenon arc lamp, high contrast, high resolution image, special fluorescent dyes used to locate molecules in specimens. Black background, bright stained specimen, no condenser required, light comes from the above specimens. Multiple fluorescent probes available, detect small quantities, molecules, can use antibody staining techniques. So these are the epifluorescent images. Location of specific molecules can be found. So here we can see immunofluorescence of tetrahymena, a ciliated protozoan. Using the fluorescent dye, we use fluorescent isothiocyanate, that is FITS in the short form, which emits green light. And above right is the cell stained with the antibody against cell membrane surface antigens, and we can compare both tetrachimer cells in these conditions. And this is the stained with hematotoxylin, and we are seeing these cells like that. So mainly it helps in the to check things with the fluorescence dye. That's how the epifluorescent microscope works out. So this is another image showing you metaphase state in the new length cells, where your cells are having these nice centrioles, centromeres is crossing, being separated apart, and then you having two different cells being generated. Then comes the differential interference contrast, that is DIC microscope that we discussed just now, also called as Normarsky optics, that resembles phase contrast but more sensitive, gives higher resolutions, and therefore we can see things in quantitative manner in this case. So we have a mercury lamp there, beam splitter, polarizing filter also, and recombinant prism at the top. So it's a very highly complicated, which gives 3D images actually. You get 3D images from the DIC microscope. An excellent resolution is there, a high numerical aperture, high contrast, and detect changes in the refractive index of specimens. So it has epifluorescence and DIC capabilities. Both things are possible in this. So these are the very fine images. So DIC is giving very fine and very exact fluorescent images that we were looking for. So DIC image is a cricket or oval showing oocytes. Here we have seen the oocytes with bulbony bodies B, and also mitochondrial clouds. So we can also see these mitochondrial clouds in these cases. And at the bottom is the three-dimensional image of the same, we can see, of the bulbony bodies. Then comes mostly whenever you will go to the lab, you will see either inverted microscope there, or simple microscope attached to the computer, or fluorescent microscope, epifluorescent microscope mostly are present, and more than that, then confocal are also quite present. So in my lab, when I was working in Germany, so we have inverted microscope, we have fluorescent microscope, which were attached to the computer, so we can take images and save them for the later. And we have confocal laser microscope, and we also had that time electron microscope also, but we were not allowed to use them because it's very advanced, and we had very small experiments at that time. So it mainly depends upon what you're looking for. So I'm telling about you each microscope. So each microscope has its own advantages. It has uh, its own importance, but what we are looking for is what is the application of it, how it could help in your creating the questions that you're looking for, to answer them. So each microscopy could really help you to answer them. So that's why a microscope, when you are doing your experiments, which type of microscope will be important to answer your questions matters a lot. So it should be of high magnifications, images will be better, the more the image better, the better will be your results, better you can answer things, better you can approach things quantitatively. Like this one, I will show you now. So these images that I'm going to show you is from the inverted microscope of the neonatal rat cardiomyocytes. So here, so I made a videos out of it. So this is a control cardiomyocytes without any addition of pharmacological agents, then 100 micromolar, then 1000 micromolar, and 10,000 micromolar. So here you can see that it's a like 4 seconds video, but it took me 24 hours to make them. So I put them, so this is the fast version of it. So what you can see is heart pumping, like cells are pumping within them, and this is nothing is happening here. and here you can see the cells start to pump slower, and here they all die. All the nucleus comes out, and there's nothing is happening. So that's the application of microscope. So that's how you can be confirmed with your results, that whatever you are looking for, whether they have any meaning or not. And this was done on the inverted microscope, all these details. Yeah. So one more image from the microscopes that I found during my PhD thesis was, this was my presentation that I gave when I was at the thesis time. So these were the images that I found. So DAPI is meant to check the nucleus. So this is a siRNA of CTGF and control siRNA. So here I was checking the fluorescence dye of different proteins CTGF, and this is a merge image of it at 20 micrometer of the length, and that's how I differentiate between the results. So I was checking that ATF6 is present in the control outside and in the knockdown. So I see the present in the inside of the nucleus. So that's how we were able to differentiate between them. So that was something about microscope. I want to show you the applications, how important they are, and how things could be really matters when we are doing with the in the field of science. So they really bring a global change to your results at the end of it. So now I go to the next part, confocal laser scanning microscope. Confocal microscope, we can say it also. So laser is focused at a plane of specimen, scans the specimen in the horizontal plane. Only light from the plane of focus reaches a detector, and scan image is digitally recorded. So image from the consecutive focal planes can be recorded, and composite or 3D images can be digitally created. So you have krypton laser in this. High resolution sharp images came out. High sensitivity can be used in the reflectiveness and fluorescence mode. Eliminate background interference. So you have biorad confocal microscope showing fluorescent labeled cells on the monitor. So like what I show you in the images from my inverted microscope, the confocal images are really better and much advanced. So these are the confocal images, and this is the bright field image. So in bright field we can't see much of the cells, but in this we can see them trichomonas immunostained with antitubulin and fluorescent isothiocyanate dye. We can see things much in better way. So now we're done with all the microscopes of simple types. Now comes the electron microscopy, that is there of two types, SEMs and TEM. So SEMs, it's the one with the fixed dehydrated specimens that are mounted on the stubs and surface are coated with the gold, palladium or rhodium. Specimen is placed in the vacuum in this case. Electron beam is strike on the samples. The electrons beams bounce off the metal coated specimen surface and collected and converted to digital image and displayed on a TV like monitor. So like this is there, this is the image of your project coated with the gold. You have electron beams coming out and they're striking it, and when we are having a checking them with the help of so if we have this ball like structure, so they came electron beam and then they could be detected on the TV like things, and we can check them and we can create a digital image later on, like they strike here, then we can check it here, they strike there and then check it there, they strike on our objective like, and they strike here, so like this. So, and they gave the 3D image at the end as CMs and with higher resolution and magnifications at the end. So SCM images, comparisons of SCMs with both images obtained using DIC microscopy, top and bottom. So both DIC microscopy and SCM reveal specimen surface topography. Much greater detail is obtained using SCM because of greater resolutions and magnifications capability. So here the SCM, so they are the 3D images. So you can see how finer the images are, how better the images looks like as compared to the normal at the bottom ones are. They got really 3D, and we can see things in much uh, in an exact manner. Then comes the transmission electron microscope. In this, we fix dehydrate our specimens, and in a resins hardened section microscopes. And then with the heavy metals such as uranium and lead, we insert the electron column in the microscopes. And the electron beam is absorbed, diffracted in the heavy metal stains, and shadows are cast onto the film of fluorescent plate at the bottom of the column. So you get not 3D images in them, but 2D. And reveals the internal cell structures are revealed, like Golgi, ER, vacuoles, all these structures. An electron beam is focused by magnetic field. So comparisons of TAM and SAM. So trichomonas are viewed with SAM and TAM here, left on the right. So note that the SCM details of external structures are visible, while in the TAM, internal structures are revealed. So left side is SAM and the right side is TAM. So TAM helps you to see the internal structures, whereas SAM helps to see the whole cells from as a 3D structure. So here you can see the Golgi body, hydrogenosomes, flagellum F here, recombinant flagellums, like this. So this is TAM images of the rat liver cells on the left and pancreatic acinar cells on the right. So this is how the left and right are being differentiated. So now comes the specimen preparations for the light microscopy, scanning electron microscopy, and transmission electron microscopy. How things are arranged in them. So this is the live mounts. For the live mounts, you view them, vital stain, and then view them. For the fixed preservatives, this is for the general for the light microscopy. You fix it, dehydrate it, infiltrate it, embed it, sections, mount it, stain it, and view it. To view organisms, tissues, or cells as in close to the natural states as possible, unstained live mounts are used. Viewing time of the live mounts is limited. Unstained specimens have low contrast. Supravital stains may be applied to provide more contrast or identify certain components, and these are stains that are not harmful to the living cells. And specimens may be preserved using chemicals such as formalin, acetic acid, ethanol, and methanol. Fixation immobilize molecules such as proteins and lipids. And then we can fix them with 100% alcohol and check our things with the paraffin and paraffins to mold them. So this is how the specimens looks like when you're cutting 5 to 10 meter of thick sections. So your vibrotome is there, vibrating to cut the sections. Specimens holder, knife holder is there, and then you can have your answers like to have your things that you're looking for. You can do it with this microtome. Fine sections of paraffin. Those who have worked in the histology lab, maybe I can show you how a microtome works likes. So I worked for 6 months in a lab, rotatory microtome sections. Maybe I can show you this one. So this is how microtomes looks like. You put the blade inside. So here what you're seeing is one of the section of organ of mice, and they are put in the paraffin wax, and that's how we see the results. They are stretched away like this. So that was about microtome machine. This is for the SCMs. You fix them, dehydrate, mount on the stubs, putter coat, and observe them. So fixations usually done by the glutaraldehyde, paraformaldehyde, osmium tetroxide. So you can use these. Then you do the dehydration. Dehydration is done to the carrying the specimens through an ascending alcohol series from 100% to the less. And then you add organic solvents, that is acetone or propylene oxide. Specimens for the SCMs may also be proceed in the critical point drying apparatus. Then specimens are mounted on the aluminum stubs using the sticky tape. Applications of one type of stub, application specimens to stubs arranged in holder. So a spotter coat the specimens with gold, palladium or rhodium in a special chamber to cover the specimens with a 10 to 20 nanometer thick metal layer. The stub is then inserted into the SEM, scanned and observed on a video display. So in this you can check them on the big SMs are really big. So in India, so I think Indian Institute of Sciences, they have these microscopes, and some IITs might have. Not all institutes have these microscopes. Not all universities have these microscopes. So systematic preparations for transmission electron microscope. So it is done with the help of fixation, dehydrate, infiltrate sections, apply section to grids, strain and observe for the TEM. So these are some molds to do so. It has different techniques to go for. You use various chemicals like alcohol, organics, solvents such as propylene oxides, and then also infiltrate the specimens with epoxy or plastic resins. An instrument called an ultramicrotome is used to section the specimens also. This is another one, microtome, showing you the sections being cutted out. And this is the grids that are inserted into the transmission electron microscope, and then they are observed in that way. So that's it from the microscope thing. So I will share this chapter with you. We have done 2 parts. So let's take a break here of around 10 minutes. We meet at 21:20. We meet at 21:20. Any questions, students, so far? So laser lights, as I discussed, to put in laser light on your specimens, and then you can check it of your interest, and that would be